Thank you to everyone attending today's webinar. Really excited to be here to share with you how MaxCyte Electroporation could potentially help you with engineering your cells and overcome conventional transfection challenges. So for today's talk, I'd just like to start with the basics and introduce transfection overall, why we deliver cargo into cells, how do we deliver that cargo into cells, and what's different about the way MaxCyte does that. We'll go over considerations for successful electroporation, including how to optimize with the MaxCyte electroporation platform, and talk about how that can help reduce timelines, establish stable expressing lines, and also go over a case study with an academic customer of ours in Japan who has a CAR T cell manufacturing process by DNA transposons. So first of all, why do we want to deliver cargo into cells? And for research, that could be to study gene function, either by exogenous overexpression of a gene or knockdown of or knockout of a gene to understand its function. It can be used for disease modeling, to produce proteins, as well as drug discovery and drug screening. There are cell and gene therapy applications such as immunotherapy. And on the manufacturing side, to produce proteins at large scale in either HEK cells or CHO cells, vaccines to manufacture viral vectors, or again, like a cell therapy product, but at a larger scale. So how do we deliver cargo into cells? So the cell membrane is a physical barrier that basically prevents the entry of large molecules into the cell. So to deliver that cargo, we need to use a physical or biological or chemical method to carry that cargo into the cell. So with the physical method, it usually involves permealizing the membrane or penetrating the membrane with mechanical approaches such as squeezing or electroporation, or chemical means by like soft detergents. A carrier-mediated method usually involves either a virus or chemical method that can either introduce the cargo by endocytosis or direct fusion with the plasma membrane. Today, I'm going to go over three of the most common approaches for delivering cargo into cells, including viral transduction, transfection by chemical methods, and electroporation. And when you're choosing your transfection method, there are a lot of considerations. For example, how easy it is to use, the efficiency that you need to get the cargo into the cell, and can that efficiency be applied over a wide range of cell types? Of course, like how that affects the cell viability, which is really important for primary cells because you're limited in the cell number that you have, the scale at how many cells that you can introduce that cargo into, and not just the scale, but scalability. So let's say you can do that at 100,000 cells. Can you do that then at 1,000,000 cells or billions of cells with the same efficiency and same cell viability? The type of cargo and the size of the cargo does determine success of cell engineering, and of course that time it takes from not just electroporating or transfecting cells or introducing the molecule, but to reach your end goal. And ultimately that determines the cost of your whole project, and there could be GMP considerations later down the road. So what is viral transduction? So viruses can be engineered to deliver cargo into cells in a process known as viral transduction. It can be a highly efficient delivery method, and there are different choices of vectors, including AAV, adenovirus, or retroviruses. Here, this image shows an example of retrovirus transduction, where a viral particle will bind to a cell surface through receptor interaction, and then it is either fused or endocytosed in the cell. You have viral genomic RNA, which is reverse transcribed to DNA and integrated into the host genome for stable expression. With retroviruses, for example, there is low transduction efficiency in resting or non-dividing cells. There's the potential for immunogenic effects and a risk for insertional mutagenicity. If you're considering GMP applications later, there are high production costs with manufacturing GMP virus and lengthy production times. There's also a safety risk to consider when handling, and there could be batch-to-batch variability with cargo size limitations. So all of these are important to think about if you're considering viral transduction for your research. In terms of transfection you can use, which is a non-viral approach to introduce foreign materials into cells. And traditionally, transfection involved mainly nucleic acid cargo delivery, such as DNA or RNA, but there are a wide range of cargo types that can be delivered into the cell, and you can see that below, from DNA plasmids all the way to mRNA or recombinant proteins or ribonucleoprotein complexes. And transfection is mostly done to modify gene expression, and whether you overexpress or downregulate that expression or stably express that gene. So one common method for transfection is chemical transfection, where you would complex your nucleic acid with a positively charged molecule, whether that's PEI, for example, and that would interact with the cell membrane for endocytosis, and then there would be a release step for the DNA to be delivered into the nucleus and then expressing mRNA and protein. So this method is cheap and readily available, but there are sometimes challenges with low efficiency and difficult to transfect cells. For example, primary cells or suspension cells. It can be difficult to optimize for higher efficiency. You are limited sometimes in the cargo diversity that can be loaded, especially if the charge is not highly negative. There is a time lag for entry and expression of the molecule, and there is batch-to-batch variation and inconsistent results to consider, as well as this may affect the membrane composition and maybe not adaptable for GMP processes. So electroporation can be a highly efficient method for delivering various types of cargo into the cell, and it basically involves an electric pulse running through two parallel plates. This temporarily introduces a pore in the cell membrane. If you look at a typical electroporation cuvette, which has both electrodes on both sides of the cells, you can electroporate multiple cells at the same time to introduce your cargo into them. So it can be if the process is optimized and you need to consider the field strength, which is the voltage applied, the pulse number, pulsing pattern, and duration of that pulse, you can get highly efficient delivery into the cell. But if you do this too strongly, you could irreversibly damage the cell membrane too. So it's really important to have a balance with all of these parameters to give you both efficiency and cell viability. Buffer composition can be important, where different instruments might have different buffers for different cell types. There's the cuvette material to consider. Aluminum, for example, creates toxic aluminum ions, which can aggregate DNA and induce cell death. So the type of cuvette materials does play a role in electroporation success. There could be limited scale depending on the size of the cuvette, and as we've talked about before, with scalability, being able not only to electroporate at 10 microliters or 100 microliters, but at higher volumes if needed for a larger number of cells. And then there are GMP considerations for this process as well. So what's different about MaxCyte? MaxCyte, you can see from our instruments on the left here, from the ATX to the GTX. The ATX is for basic research, but can still electroporate up to 700 million cells. The STX and GTX has this peristaltic pump, which can electroporate up to 20 billion cells. The STX is for bioproduction to produce proteins and viral vectors, whereas the GTX is our say GMP compliant platform. So with MaxCyte, you have one gentle electroporation buffer we have proprietary, we call them processing assemblies, you may know them as cuvettes, um, which have non-toxic electrodes. We have a number of pre-optimized electroporation protocols that work for various immortalized cell lines as well as primary cells. We have a seamless scalability that you can electroporate from 100,000 cells all the way up to 20 billion cells. And we can get molecules into cells that are more challenging traditionally. In addition to that, we have a team of field application scientists, about 20 FAS globally, that are specialized in different areas ranging from protein production to um, immunotherapy for supporting our customers. We have regulatory support for GMP manufacturing, and we have an internal R&D team too that has a lot of knowledge about electroporation. So our platform is also GMP ready. So just going on to preloaded electroporation protocols on the MaxCyte instruments, as I mentioned in the last slide, we have pre-optimized protocols for immortalized mammalian cells, including common ones like CHO and HEC293, or cancer cell lines such as K562 and A549. We have a number of optimized protocols for hematopoietic cells, including HSCs, activated and resting T cells, and NK cells. We also have protocols optimized for human-induced pluripotent stem cells and difficult-to-transfect keratinocytes. Also, protein production customers using insect cell lines, we have protocols optimized for insect cell transfection. And in terms of if your cell type is not on this list, we have 10 general optimization protocols from low to high energy, which can be used for new cell types and applications. This slide is just showing an example of how we can efficiently introduce GFP DNA into common cell types like CHO and K562, and more challenging cell types such as primary neurons, skeletal muscle cells, adipose-derived stem cells, and iPS-derived motor neurons. Here we show MaxCyte's processing assemblies, which basically enable electroporation from volumes as small as 15 microliters up to 100 milliliters. So in terms of how many cells you can electroporate, you can electroporate as low as 75 to 100,000 cells, all the way up to 20 billion cells. And usually optimization occurs at the smaller scale with multi-well processing assemblies, where you would optimize the cell density, the cargo concentration like mRNA or DNA, and then the electroporation protocol. Once you optimize those parameters, you would simply scale up the volume for seamless scalability. So this is an example of that seamless scalability and data generated by our in-house team. So a master mix of K562 cells were mixed with GFP DNA plasmid, and then the same master mix was added to all of the processing assemblies you see here, including each well of the multi-well processing assemblies. And essentially you can see GFP expression being very consistent from 25 microliters all the way up to 10 mLs in our flow electroporation processing assembly. Basically, so you can use the same cell density, DNA concentration, like I mentioned, and the same electroporation protocol from small to large scale to get the same efficiency. This video is an example of how our flow electroporation technology works. So the cells and loading agent are loaded into this sample bag on the left-hand side. A peristaltic pump drives a defined volume of the cells into the electroporation chamber. The cells are electroporated and then flowed out into a collection bag. The process is pretty rapid. If you electroporate 100 milliliters of cells, which is about 20 billion cells, this process takes less than 20 minutes. And while this video shows kind of a manual loading and harvest of the cells, it can be a closed process where you can bioweld the tubing to instrumentation upstream or culture vessels downstream of this electroporation process. So usually our workflow looks like this, where you would culture your cells wash them and resuspend in MaxCyte electroporation buffer at a high cell density, 100 to 200 million cells per ml. You would mix with your loading agent, whether that's DNA, RNA, or CRISPR RNP, and then load that into the appropriate processing assembly, depending on how many cells you need for your process. Electroporate, and after electroporation, we usually have a recovery step for 30 to 40 minutes to allow the cell membrane to recover. Afterwards, you would culture in culture media and then analyze the cells at the indicated time points. So, in terms of what kind of molecules you can express any type of molecule with MaxCyte electroporation, including ion channels and GPCRs, a variety of different genome editing tools, of course, CRISPR-Cas9, more recently, base editing and prime editors for protein production, such as antibody production or viral vector production with lentivirus AAV or VLPs. And also, you can use the MaxCyte platform for reporter assays and transcription factors. This slide shows an example of how MaxCyte combines high efficiency with cell viability. So on the left are images of GFP expression and bright field images of human iPS cells after MaxCyte electroporation compared with two other platforms. If you look at the quantification of this by flow cytometry, you can see that both DNA GFP and mRNA GFP, the expression is nearly 100%, whereas with another platform, you might have less efficient delivery. And with another platform, you might have high efficiency, but then you sacrifice the cell viability here on the right. So MaxCyte kind of gives the best of both efficiency and cell viability, and that's enabled through the benefits I mentioned earlier with pre-optimized electroporation protocols, our processing assemblies, and our buffer. And then I'd like to go over next considerations for successful electroporation. Okay, so next I'd like to go over how to set goals before you start. And you need to consider how long your workflow is going to take, not just the transfection portion, but all the way to achieving those final results. Are you taking the right engineering approach? Does it have to be done with DNA? Could you do it with RNA, for example? And then maybe there's another approach that you didn't consider for cell engineering. Is long-term and constitutive gene expression needed, or can you do it transiently? And then what are the critical quality attributes for those transfected cells after transfection? So how MaxCyte can help is with shortening timelines. And if, for example, you have an inefficient transfection method, and this is an example of a customer that we had, which had an inefficient transfection method, which resulted in them having to enrich and select their cells. This caused the cells to have a longer recovery period and expansion period. So this was up to 5 weeks, for example. And if you use a viral transduction approach, you could transduce and expand the cells in a shorter time, but there is an additional 1 to 2 weeks where you have to produce the virus, purify it, and titrate it before you can actually transduce the cells. With MaxCyte electroporation, once you have your cargo that you would like to load to the cell, you can electroporate and expand the cells right away to shorten your timelines for higher success rates. So critical factors for successful electroporation with MaxCyte, you need to consider the type of cells you're using, the quality of those cells too. You need high cell viability, a low passage number with cell lines, and to ensure that the cells are in log growth phase before electroporation. After electroporation, you need to recover the cells, and you can vary the cell seeding density or plating density. If applicable, you can also cryopreserve the cells without losing function. And the type of cargo that you put in cells is really important in terms of the quality. If it's like DNA that you're purifying, it needs to be endotoxin free. How much DNA or mRNA you put in, the size of that cargo, as well as the charge stability and how toxic it could potentially be to the cells. So this slide shows an example of how to optimize the DNA concentration during electroporation. So this is in CHO K1 cells. What you can see over here is from a low amount of DNA from 50 micrograms per ml all the way to 400 micrograms per ml, you do increase the efficiency as you increase the amount of DNA. That also results in higher mean fluorescence intensity. But as you go higher in the DNA concentration, there is a drop in cell viability. And while these CHO-K1 cells can tolerate the higher amount of DNA, some other cell types might not be able to. So another consideration is the correct promoter to use. So this is an example in human iPS cells with a GFP DNA plasmid with a CMV promoter versus an EF1 alpha promoter. So you can see with the CMV promoter, which does get silenced in human iPSCs, with 200 micrograms per ml of DNA, you're getting about 32% expression. You can increase the amount of DNA to get nearly 80% expression. However, if you just simply change the promoter, you could achieve almost 100% expression with the lower amount of DNA without having to go to a higher amount of DNA and potentially sacrificing the cell viability. So this slide shows how the size of the DNA affects the transfection efficiency. And again, this is human iPS cells, but basically what you can see is from 9 to 10 kb plasmid, you can get over 90% efficiency. As the plasmid size increases, the efficiency does decrease. Now, this is really dependent on the type of cell. We've had situations where customers can deliver a 100 kb DNA into other cell types. With iPS cells, you can still get up to a 10 kb plasmid in before the efficiency starts going down. With primary cells, it's much more challenging to get larger DNA plasmids of 9 kb in. If it's like T cells, for example, you might be starting at 20 to 30% instead of over 90%. So just wanted to point out how the size of the DNA plasmid could affect the transfection efficiency. Okay, so to further discuss that point about how DNA size limits transfection efficiency, this is an example of data generated by our in-house team in human macrophages with 3 DNA plasmids of different sizes ranging from 2.5, 4.5, or 9.2 kilobases. And when delivered at 2 different energies, either a low energy or a high energy, and 2 different DNA concentrations, either low or high, you can see that the smaller DNA plasmid at 2.5 kb can get in with pretty high efficiency even with the lower energy. And as you increase in DNA size, the efficiency does decrease. You can compensate for the decrease in efficiency by increasing the electroporation energy here. So you can see that even with the medium-sized plasmid, now you're getting in at over 80%. The larger plasmid still has challenges of getting in, although you can get in at 60% here. The other consideration too is that the large DNA plasmid has a bigger impact on the cell viability. So you can see even at the lower energy with the higher DNA, you're getting less than 20% cell viability. And then in terms of higher energy with higher DNA, the cell viability drops even further. So you do need to balance the size of the DNA plasmid, the concentration of DNA, and the energy that you're delivering. This slide shows an example of gamma delta T cells from a collaborator at the National Cancer Center in Japan, where they delivered either mRNA GFP or DNA GFP. And what you can see here is that the GFP mRNA has high efficiency that's sustained for about 96 hours after transfection, whereas DNA with the lower DNA compared to the higher DNA concentrations, you can get higher transfection efficiency with more DNA. However, there's a cost of cell viability. mRNA has higher cell viability when used compared to DNA plasmids, especially with the higher DNA concentration, which takes a few days to recover. But the other consideration too is that cell expansion, where cell expansion is much lower when you're delivering DNA. So in this case, if there's a way to deliver mRNA instead of DNA, that would be preferred for this cell type. And kind of uh, to continue that line of thought, if you're using genome editing tools like CRISPR-Cas9, you can deliver them as mRNA or ribonucleoprotein complexes. This is an example showing mRNA Cas9 with guide RNA in Jurkat cells. So you can see with 3 different concentrations of guide RNA, you can get over 90% knockout efficiency of the track locus in Jurkat cells. If you use the ribonucleoprotein complex, you can get the same efficiency as well. In primary T cells, you can also use ribonucleoprotein complexes to get high knockout efficiency of 90%. And in all conditions, the cell viability was over 90%. So really, depending on the type of cargo you put in, that can also determine your electroporation success criteria. So how to simplify the process of optimization with MaxCyte electroporation? So you can use MaxCyte electroporation for cell-based assay development because you can transfect any cell and any molecule with a range of cells up to 20 billion cells. You can develop and optimize the assay and any cell type you need to, and readily adapt to existing assays for new targets. So scale up allows potential cryopreservation of the cells, so you can minimize the drift in expression when you're passaging cells. You can use this for high throughput screening as well as delivering consistent performance day to day. So this is an example of multi-subunit expression in cells where 4 plasmids expressing 3 subunits and an inward rectifier were delivered into CHO cells, and then you can see the reconstitution of the ion channel by this fluorescence signal. If you add an antagonist, the antagonist can inhibit the signal of the ion channel. So this data is just to demonstrate that multiple plasmids can be introduced to reconstitute ion channel function, and you can use it for drug screening as well. You can also vary the DNA concentration during electroporation, where you can see here that the in U2OS cells transfected with a reporter plasmid, you can modify the reporter expression based on how much DNA you put in the cell. So low amounts of DNA compared to a higher amount of DNA, you can control the expression of the reporter. And in terms of varying the DNA concentration in combination with cell plating, this is an example of HEC293H cells where you transfect with a NF-kappa beta expression plasmid and a luciferase reporter. So with a lower amount of DNA and higher amount of DNA, you're getting higher expression of luciferase with the higher DNA. And you can also add more cells for plating per well to increase that signal if necessary. You can also use MaxCyte for protein production to essentially produce milligrams to multiple grams of protein within days of electroporation, instead of using instead of establishing a stable cell line, which takes time. This is an example of diabody production in CHO-S cells, comparing lipid transfection with the MaxCyte STX. So you can see that MaxCyte basically provides or yields 20 times higher titers than lipid transfection, and also a higher quality protein product. In terms of how else you can use MaxCyte large-scale electroporation of up to 20 billion cells, this is a media strategy you can use for transfected cells. So you can bulk electroporate cells and then split them into multiple flasks to test different media and feed combinations to analyze which combination works best for generating the best titers. So you can see here, this is using 14 different combinations of media and feed. And in combination 14, by day 12, you're getting nearly 7 times higher expression than with other combinations. Also, in terms of shortening timelines for generating transient materials, you can see here that with small scale electroporation versus large scale electroporation, you can get similar titers of about 1.2 grams per liter. If you're doing that at small scale, that could result in 21 milligrams. And simply scaling up that process from 20 milliliters to 2.8 liters of culture, you can obtain 3.5 grams. So really that scalability allows you to generate enough materials depending on what those downstream analytical processes involve. So next, I'd like to go over how MaxCyte scalable electroporation can reduce timelines. And in terms of assay-ready cells, you can electroporate and cryopreserve the cells with the desired cell number in either plate or tube format and the desired concentration. Usually after thawing the cells, within 24 hours, they're ready to use for drug screening, for example. And it's a convenient and time savings method for assay development. And you can also use this assay-ready cells with stable lines or transiently transfected cells for off-the-shelf targeting with cell types and certain targets. Okay, so here we're looking at rapid assay optimization with assay-ready cells. So you have the option after MaxCyte electroporation to either plate the cells without freezing and analyze perhaps screening at 2 or 3 different time points, or you can freeze the cells after electroporation. You can freeze them either immediately after or 1 to 2 hours after electroporation, or even culture the cells for up to 48 hours before freezing them. This is an example in Jurkat cells with a luciferase reporter and a nuclear receptor expression plasmid, where you can see freshly electroporated cells and cryopreserved cells basically have the same IC50 for compound inhibition, whether using fresh or cryopreserved cells. So if you can actually electroporate a large number of cells, cryopreserve them, and then be able to take out that frozen aliquot for screening to give consistent results, that will ultimately save time in the end, and you'll get similar performance to freshly electroporated cells. These are publication examples of transiently transfected assay-ready cells, from ubiquitin ligase drug discovery to high-throughput screening of ion channel variants, and also including drug discovery with SARS-CoV-2. And then in terms of approaches for stable expression, there are multiple ways to do this. With protein production, as I mentioned before, if you can electroporate a large number of cells, for example, CHO cells, you can generate transient materials. At the same time, you can take a portion of those cells to produce either your stable pool, mini pools, or single cell clones to kind of save time. So this is an example of rapid bulk pool development using a random integration method. So cells are transfected and then selected with an antibiotic selection. So in blue, you see this is MaxCyte electroporation with 4 different antibody constructs, and compared to in purple chemical transfection with the same 4 constructs. So you can see with electroporation that the pools recover much faster from selection than chemical transfection. In some cases with chemical transfection, they don't recover at all. So just basically increasing the time, shortening the time needed to obtain that stable bulk pool. And then in addition to random integration, you can use a DNA transposon based method for gene delivery. So this is data generated by our in-house team, where you can use K562 cells with a DNA transposon and an mRNA transposase. So on the left-hand side here, you see increasing amounts of the DNA transposon at day 17 after electroporation, and then also increasing amounts of mRNA transposase. So the transposase will mediate the the um, the integration of the transposon into the genome. So you can see after 17 days, even with the highest concentration of DNA transposon, you don't have any integration of the DNA transposon. Whereas if as you increase the amount of transposase, that you can basically get higher levels of cells with integrated copy numbers of DNA. And this slide is just quantifying the copy numbers of DNA in the cells at day 0 versus day 17. So you can see here with increasing amounts of DNA, whether that's without transposase or with transposase, you can increase the number of copies of DNA being delivered into the cell up to 800 to 1,000 copies per cell. However, after 17 days, when there's no transposase delivered to mediate the integration of that DNA, you don't have any copies of integrated DNA in the cell. And then if you have the transposase, you can control the copy number by the amount of DNA transposon that you're co-transfecting. So with the lower DNA, you'll get lower copy numbers up to 2 copies per cell. Whereas if you increase the amount of DNA transposon, you can get over 15 copies per cell. And in addition to random integration and DNA transposons, you can use a CRISPR-Cas9 mediated approach for CAR knock-in with either DNA plasmids or nanoplasmids, which are smaller plasmids which have had the basically the DNA backbone size reduced as donor templates. If you combine that with a Cas9 targeting sequence, which basically binds Cas9 for nuclear import of that DNA donor template, you can get high efficiency of knock-in. This is in activated T cells at the track locus. So this is showing viable T cells, the percent viable T cells out of total gated population in flow cytometry. So no electroporation versus 3 different concentrations of the homology-directed repair template. From 50 micrograms per ml up to 200 micrograms per ml, you can see that the viable cell populations is pretty similar to the non-electroporated. The track knockout efficiencies can reach as high as 90%. And in terms of the knock-in efficiencies, if you have the CTS sequence on the DNA donor template, there is higher efficiency of knock-in with the nanoplasmid versus the normal size plasmid. And you can see that basically with the smaller nanoplasmid and the medium concentration of DNA donor template, you can get the highest knock-in efficiency of about 35%. So really this data is to show that if you can optimize your DNA donor template, you can significantly improve the knock-in efficiency, and smaller HDR templates help with that knock-in. So lastly, I'd like to go over a case study of an academic customer we have in Japan manufacturing a CAR T cell product for using piggyback DNA transposons. So this is work from Shigeki Yagu's lab, who was previously at Kyoto Prefectural University of Medicine and is now at Shinshu University. He was trying to develop CAR T cells with a non-exhausted memory cell T cell phenotype and sustained anti-tumor activity. So to develop this process, he was targeting or trying to treat rhabdomyosarcoma in pediatric tumors, and he wanted to choose a non-viral method which was more affordable, faster, and hopefully highly more functional than viral delivery. So he did optimize piggyback DNA transposon and transposase plasmids and PBMCs using the MaxCyte platform to generate EPH before CAR T cells for both in vitro and in vivo activity. One of the reasons why Shigeki came to us was because he needed help with optimization using electroporation. So we worked with him on new electroporation protocols, and ultimately we found a protocol that enabled pretty good efficiency. This is just showing early optimization work with 2 different DNA concentrations. Of course, if you increase the amount of DNA, you'll get higher expression of GFP, but the cell viability drops. So one way Shigeki overcame this issue was by increasing the cell density during electroporation from 100 million cells all the way to 800 million cells per ml. And he found that actually increasing the cell density increased the transgene expression and improve the cell viability. This also reduces the amount of DNA he had to use because he can use a smaller volume for electroporation. So his process involved electroporating PBMCs with a DNA plasmid for the transposase and a transposon, either expressing CD19 CAR or EPH before CAR T. And then he also electroporated autologous feeder cells to present the antigen. So he would irradiate the feeder cells, cold culture with the CAR T cells, and then expand the cells for about 14 days. After that expansion process, with 2 different CAR T cells, CD19 and EPH before CAR, he was able to get 50% and 90% expression of the CAR respectively. And in terms of the memory phenotype, they had majority stem cell memory phenotype with low PD-1 exhaustion markers. And how that affected the killing activity of the cells, he co-cultured EPH before CAR T cells with target cancer cells and subjected the CAR T cells to 3 different subsequent rounds of killing assays. And he found that the cells could retain killing activity up to 3 challenges with the tumor cells. So also demonstrating that the cells did not have an exhaustive exhaustion phenotype. In terms of testing the CAR T cells in vivo, this is a non-specific anti-CD19 CAR T cells and a specific EPH before CAR T cells targeting tumor cells which express luciferase. So the non-specific CAR T cells resulted in the tumor expanding, indicated by the increasing luciferase expression. And then with the specific CAR T cells, it did suppress luciferase expression in the mice. And ultimately, Shigeki has been able to scale up this process, and he's close to clinical trials. But this data is just to show how closely we work with customers in order to ensure his success, because he wasn't able to successfully electroporate PBMCs initially until we worked with them on a new electroporation protocol. So in summary, the cargo type and design, the purity and concentration of the cargo can impact transfection efficiency. Other parameters that affect transfection efficiency success are the cell health, the concentration and density of the cells, and hopefully we showed today that MaxCyte electroporation could enable high efficiency of multiple loading agents in cargo, including DNA, RNA, and protein within a wide range of cells to reduce your timelines and increase experimental success. You can leverage large-scale transient transfection for cell-based assays and protein production. And then there are multiple ways to establish stable cells through random integration or DNA transposons or specific CRISPR-Cas9 mediated knock-in approach with DNA donor templates. And lastly, our example with our academic customers showing how MaxCyte worked closely with them to ensure their experimental success.